Dear students, now we are going to discuss photosynthesis in higher plants. So here, what are the higher plants? Particularly angiosperms, some extent gymnosperms also. If you go back to plant kingdom, algae, bryophyta, pteridophyta, gymnosperms, and angiosperms. So out of all these, primitive is algae definitely. So advanced angiosperms. Okay. So this chapter mainly focusing on mechanism of photosynthesis in higher plants and maximum extent all the events except few cases are similar for all photoautotrophic organisms, not only restricted to plants. So all these events maximum extent present in almost all photoautotrophs. So it is a part of metabolism. Metabolism is, what is metabolism? A sum of all biochemical reactions in organisms, collectively called as a metabolism. In metabolism, there are two types of pathways, anabolic and catabolic pathways. So photosynthesis is a anabolism. So anabolism means a constructive reaction. So during this reaction or this metabolic pathway, simple inorganic components, simple inorganic components. What are the simple inorganic components? H2O, majorly, mainly I am taking CO2, are converted into complex organic compounds, particularly carbohydrates. H2O and CO2 with less amount of energy. They need a lot of energy to convert themselves into carbohydrate. So photosynthesis is a energy consuming reaction. Light energy is required. And hence the name, it is a photochemical reaction. Photochemical. What is this photochemical? Physical energy, light is required. Light is ultimate source of any organism. To perform some work, energy is required. So definitely that energy is coming from sunlight only. That is ultimate source of energy. So photochemical, photo, light, and chemical. What are the chemicals produced? Here, carbohydrates. Carbohydrates. So almost all heterotrophic organisms, those which are not able to perform photosynthesis, according to statement given, all organisms are dependent on plants, all animals, and most of the other organisms are dependent on photosynthesis. So that is a prime source of two things majorly. One is food and second one is oxygen. So all organisms except some photoautotrophs, rest of the organisms are dependent on plants. So what about the importance of photosynthesis? That photosynthesis is concerned with what are the services, what are the end products or byproducts on which all these organisms are dependent. One, food, and second one is oxygen. So without oxygen, obligate aerobes. Obligate aerobe means organism which is able to survive in the presence of oxygen only. So out of a total amount of oxygen available in this ecosystem, the maximum amount of oxygen is from oxygenic photosynthesis. Oxygenic means oxygen producing photosynthesis. Oxygenic photosynthesis. So the importance of photosynthesis is not only food, but also oxygen. Okay. So it is a photochemical reaction, right? So during light reaction, sunlight, radiant energy is converted into chemical energy, ATP and NADPHH plus, right? So that's about the introduction part of photosynthesis. Now, what about early experiments? What do we know about photosynthesis? What do we know about photosynthesis? So we have to study various experiments with the help of which we can understand the mechanism of photosynthesis and what are the factors required for photosynthesis. So according to our knowledge, according to 8th or 9th or 10th class knowledge, what are the factors which are required for effective photosynthesis? One, chlorophyll, that is a plant factor. Light, environmental factor. Water required. Okay, so various inorganic nutrients required. So with the help of all these raw materials, the plant is able to reduce the C of CO2 into carbohydrate. So to understand that mechanism, we should focus on early experiments. So here, first one, chlorophyll is essential for photosynthesis. Chlorophyll. Chlorophyll required for photosynthesis. So what are the photosynthetic parts present in plants, higher plants? Okay, so particularly the major part is definitely leaf. In addition to leaf, certain areas where chloroplast is present, definitely, or chlorophyll present, they are able to perform photosynthesis. For example, roots with chlorophyll, photosynthetic roots, teniophyllum. Teniophyllum, if you go back to morphology, under the heading modified roots, teniophyllum, photosynthetic roots. So, but major part is definitely leaf. So what about how to demonstrate the importance of chlorophyll? Okay, so the given one is variegated leaf. Variegated leaf. What is this variegated leaf? So variegated means with different colors. Variegated leaves. Variegated leaf. A leaf with isolated patches of colors, different colors. Okay, so for example, in this area, a red color area, for example, only. This is one type of color, isolated colors. Next, green color, for example, only. And some other color. So this is called as a variegated leaf, one type of mosaic leaf, mosaic. with a different isolated colors, with different colors, simple. Okay, so here I'm taking this part. Only certain areas are in green. Okay, so except red colored one, black colored one, remaining areas are green, for example. So green means definitely that is with a chlorophyll. Here we are going to demonstrate that chlorophyll is essential for photosynthesis. So that's why they took variegated leaf. Actually, in NCERT, the given statement is 2 leaves. We'll discuss that one also. The given statement, 2 leaves. One of them is a variegated leaf. First one, variegated leaf. So now this leaf is exposed to or irradiated with light. Okay, so after certain period of time, now this leaf is treated with iodine. Iodine. Go back to biomolecules. Iodine test is for starch or not? Starch is with a secondary helical structure. Go back to biomolecules. First year part. Secondary helical structure, spring-like structure with helix coils are present. So iodine is held up in the helixes of starch. Okay, so iodine reacts with the starch. Then what about the color? That is well-known thing. So iodine is used to test starch. So blue in color, in this area, blue colored region, blue colored region. Rest of the area, white color. So what that indicates? In that area, chlorophyll is present. So chlorophyll is mandatory, essential for photosynthesis. In the rest of the area, no blue color. Rest of the area is white color or colorless. So if blue color is appeared in certain areas, definitely their chlorophyll was present. So in the rest of the areas, no blue color means no chlorophyll, no photosynthesis. That is about a variegated leaf. Next, demonstration of light. So light is essential for sunlight. Light essential for photosynthesis. Light is essential for photosynthesis. Okay, so here given one. This is a leaf. Half of the leaf is covered with a black paper. So covered with a black paper. In 8th class, you studied this one with the heading light screener, light screen experiment. But here they explain differently. So many experiments are there. But ultimately, the goal is to demonstrate the concerned one. So this is a black paper. So black paper prevents entry of sunlight or not? Definitely. So this area is will not be exposed. So the purpose of black paper is it is a light screener. Light screener. Okay. So now it is exposed to light. Okay. So after that, after a certain period of time, now it is subjected to iodine test. So what about the appearance of this leaf? Okay, so half of the part, for example, only. So this one is blue in color and this one is colorless. So according to that experiment, basically actually the leaf is having chlorophyll here, chlorophyll here. Chlorophyll is not a limiting factor. So entire leaf was green in color, completely green in color. Okay, so now half of the green colored part blocked by, covered by black paper to prevent entry of light. And now tested with iodine. So blue color region is present at the base of the leaf, not in the area where it was covered by black paper. So, but in your textbook, the given statement to demonstrate the importance of chlorophyll and light, green part, actually given statement, green part and light, two types of leaves are required. What are those two types of leaves? In one sentence, they explained both the experiments. Importance of green part, chlorophyll, importance of light in photosynthesis. So kind one, kind two, two kinds of leaves in this experiment. So actually two experiments are there. One is for chlorophyll, the other is for importance of light for photosynthesis. Okay, so factor one, factor two. Third factor, what about the third factor? Most important limiting factor, that is CO2, carbon dioxide. That is a moles half-leaf experiment, third one. Third experiment, moles half-leaf experiment to demonstrate the importance of CO2. Okay, so this is a well-watered potted plant. Okay, so take one leaf. So half of the leaf, upper part of the leaf. So now it is in green color. Chlorophyll is not a limiting factor. Okay, so we are going to limit some factor. What is that factor? We will see. So upper part of the leaf is inserted into a test tube with the help of a split cork. Split cork. So upper half of the leaf is inserted into test tube, and the test tube is with this one is KOH soaked cotton. Okay, so cotton. Purpose of cotton is to hold KOH. Purpose of cotton is to hold KOH in that area. Okay, right? So now it is exposed to light. Okay, so after that period, that leaf is subjected to test, iodine test. After that, iodine test. So what about the basal part? This is basal part, upper part. So basal one is a blue color and this one is colorless. So in this experiment, all means in this area, all factors, light available, CO2 available, chlorophyll available, all are available. In the test tube, all are available except CO2. So what is the purpose of KOH? KOH is oxygen, sorry, CO2, sorry, CO2 scavenger. CO2 scavenger. So KOH is able to absorb carbon dioxide present in the test tube. Definitely, they will ask one question. The purpose of cotton is to hold KOH. And what is the objective of KOH? To observe CO2. So within the test tube, the limiting factor is CO2. Rest of the factors, light, chlorophyll, water, everything is available, supplying from the pot. So limiting factor, that is able to reduce, limiting means less available. So limiting factor is CO2. So according to this experiment, CO2 is important for photosynthesis. Okay. Next, fourth experiment. That is Joseph Priestley's experiment. Joseph Priestley's experiment. So what about the statements given by Priestley during his experiment at the end of an experiment? What about the statements given by Priestley? Air. Air is essential for plant growth and development. So air is very essential for plant growth and development. And he discovered or identified oxygen. Oxygen was discovered by Joseph Priestley. And according to his experiment, what about the statement given by Priestley? Green plants are plants, green plants restore air removed by breathing animals So they are able to restore air which is removed by breathing animals. Breathing animals means they are taking oxygen. Okay, so that oxygen is depleted by breathing animals, restored by green plants. Right? So what about the experiment of Priestley? So this one is a bell jar experiment, not for transpiration, it is for photosynthesis. Okay, so this one is a candle and a rat. Here a mouse, I'm taking a rat or mouse. Here mouse is present. This one is mouse. Or you can write rat. Okay, so that is rat. So they continued, means burning of a candle continued for some extent. And mouse survived up to certain extent only. So after certain period of time, what about a candle? Extinguished. That a candle was extinguished, stopped. And what about a rat? Died. Rat died because of suffocation. Lack of something, whatever it is. So go back to third statement. Green plants restore air removed by breathing animals. Or burning candle. You can use burning candle also. So in this case, candle stopped burning. Rat died. Reason, in this case, the limiting factor for both the objects, living and non-living objects, is lack of oxygen. So no oxygen. Because of no oxygen, burning of candle stopped. And a breathing animal, because of no breathing, that was died. The second experiment. Now, into the bell jar, an angiospermic plant was introduced. That is mint plant, mint plant. Okay, so mint is an angiospermic plant. And a candle. This one is candle towards one side, no problem. Candle and a rat. Okay, so what happened? Continuity of a burning candle and a mouse survived. That is because of oxygenic photosynthesis. Mint is able to perform oxygenic photosynthesis. What is oxygenic photosynthesis? Oxygen evolving photosynthesis. So third statement, green plants restore air removed by breathing animals and burning candle. So how to rekindle? How to rekindle this one without disturbing the experiment with the help of lens. With the help of lens and sunlight, definitely we can ignite the wick part of candle. So how to rekindle or lighten that one again without disturbing the experiment with the help of a lens. Okay, with the help of lens, divert the sunlight onto the wick. Definitely it can burn. So that's about a fourth experiment. Next. Okay, so fifth one, John Ingenhouse's experiment. John Ingenhouse. It was given in 10th class. John Ingenhouse experiment. John Ingenhouse experiment. That experiment is a hydrilla funnel experiment. This experiment is well known to you. Okay, so what is the objective of Ingenhouse? Actually, the objective of Ingenhouse is to study importance of or essential role of importance of sunlight. Importance of sunlight in photosynthesis. Okay, so the given statement by Ingenhouse, he demonstrated that light plays an important role in a particular function of plants, which means that function is somehow, means some extent, able to purify the gas or environment which is fouled by or damaged by breathing animals and a burning lamp or candle. So what was the main objective of Ingenhouse? To study the importance of sunlight in a particular process, whatever it is. Nowadays, we are calling that one as photosynthesis. In a process, by means of that, the plant is able to purify the gas fouled by breathing animals and burning candles. That was the main object. So what about the experiment? Hydrilla funnel experiment. So beaker filled with water. And take a funnel into which already hydrilla inserted. Hydrilla plant. Okay, so hydrilla. So hydrilla is an angiosperm. Hydrilla is a angiosperm. Flowering plant. Submerged, suspended hydrophyte. Submerged. Go back to ecology. And the heading you studied various types of hydrophytes. Free floating hydrophytes. Like that. Submerged, suspended hydrophyte. So hydrilla is submerged. inside the water, suspended within the water only, not in contact with soil and atmosphere. Okay, and covered with a test tube. Okay, so now this experiment is kept in sunlight. Kept in sunlight. No, so now light is available in the light. And what he observed, that is well known to evolution of bubbles. Bubbles. So what is that bubble? Oxygen. In the absence of light. So he did two things. One, that experiment was kept in the area where sunlight is available. If sunlight is available, he observed evolution of bubbles from the cut end of so-called plants. If it is kept within dark area or in the absence of sunlight, where there is no sunlight, there was no evolution of bubbles. Now, go back to the statement given by Engine House. Importance of light in a reaction of plants. That reaction is able to purify the gas, atmospheric gas, which is fouled by, damaged by breathing animals and burning candles. So that's about a hydrilla funnel experiment. Now, brief description for hydrilla. Hydrilla is an angiospermic plant. Hydrophyte, submerged, suspended hydrophyte, able to perform oxygenic photosynthesis. Oxygenic photosynthesis. So that is about a hydrilla funnel experiment. So here importance of light in photosynthesis, by means of which plants are able to purify the gas fouled by breathing animals and burning candle. Next experiment. Next one is a TW Engelmann experiment. TW Engelmann experiment. Okay, so this is very, very important. This experiment is very, very important. So this is a source of light, sunlight. Okay, so out of a electromagnetic spectrum of light with a different wavelengths of light. So from that he selected only visible light. Visible light. Visible light. That is with 7 colors. Okay, VGR. So now visible light. Okay, so now visible light is passed to prism. Prism experiment. Prism. That prism is well known. So once if this visible light is passing through the prism, now it is divided into spectral components. What are they? One by one. 1, 2, 3, 4, 5, 6, 7 with GR. With GR, wait. Okay, 1, 2, 3, 4, 5, 6, 7. 7 plus WIPGR. Okay, so these are the components of visible light. Okay, right? So now test tube. Test tube with water. Okay, now into which he introduced a plant. That plant is very, very important. What is that plant? That plant is Cladophora. Go back to algae. Cladophora. Okay, so this plant is a Cladophora. Okay, come back here. Cladophora. What is Cladophora? Cladophora is green alga. Green alga. That is chlorophyll member. Go back to plant A. Chlorophyll. Okay, so what about the characteristics of Cladophora? This is very, very important. Cladophora is multicellular. Plant body, multicellular. Many cells. Multicellular, unbranched. Unbranched. Uniseriate, one row. Uniseriate. Gametophyte. Gametophyte. Dominant phase is haploid gametophyte. Okay, so that is able to perform oxygenic photosynthesis. Okay, now very, very important thing. Now a suspension of means certain number of aerobic bacteria. Aerobic bacteria. Bacteria which are able to perform aerobic respiration. A suspension of means a sample with aerobic bacteria. Sample with aerobic bacteria introduced into test tube with water and Cladophora. Okay, so this area of test tube means here Cladophora is exposed to violet. In this area, indigo. In this area, blue. In this area, green. In this area, yellow. In this area, orange. In this area, red. So along the length of a Cladophora plant body, WIPGR concerned wavelengths of light available. Okay, and what he identified, accumulation of more number of bacteria. Where more number of bacteria in the red light, more number of bacteria here I'm taking more number of bacteria accumulated here. Next, blue region, blue light, more number of bacteria. In the rest of the area, absent or less available. Okay, so what are those areas? This one is blue region and this one is red. Red means red available, blue available regions. So here, what is the type of respiration performed by bacteria? Aerobic respiration. For aerobic respiration, oxygen required or not? So during, once they introduced into so-called test tube, they are checking for oxygen. They are moving towards area where O2 is available, dissolved oxygen, DO, dissolved oxygen. So dissolved oxygen, DO is more here. DO, dissolved oxygen. Dissolved oxygen. So in blue region, more amount of oxygen. Red region, more amount of oxygen. Okay, that is because of how he is saying What is the reason to say more oxygen evolved here, more oxygen evolved here? On the base of number of bacteria. Okay, right? And he prepared, on the base of this experiment, he prepared action spectrum. Action spectrum. What is action spectrum? Action spectrum. Action spectrum means, what is action spectrum? A graph that shows photosynthetic ability of various pigments at different wavelengths of light. What is a action spectrum? Action spectrum means a graph that shows photosynthetic ability of pigments at different wavelengths of light. Action spectrum. Okay, so he prepared a first action spectrum was prepared by Engelmann on the base of evolution of wood. On the base of evolution of wood. For example, go back to complete equation of oxygenic photosynthesis. 6CO2 plus 12H2O, H nu chlorophyll gives rise 1 glucose molecule plus 6H2O plus 6O2. How many O2 molecules evolved? 6 O2 molecules. Means 12 half O2s. Okay, so in that equation, photosynthetic equation, 6CO2 plus 12H2O, light chlorophyll, 1 glucose molecule, 6H2O plus 6O2. How many O2 molecules? 6. If number of O2 molecules are given, definitely we can assess what about the number of glucose molecule. Ratio between glucose molecules and O2 molecules in this case is 1:6. So on the base of number of oxygen molecules evolved, definitely one can able to calculate the number of glucose molecules. So what is action spectrum? A graph that explains the photosynthetic efficiency of a pigment, that may be chlorophyll A or B or carotenoid, at different wavelengths of light. That is action spectrum. That is almost similar to absorption spectrum of chlorophyll A. What is absorption spectrum? Absorption spectrum means a graph that shows photosynthetic absorption, means absorption of light. What is that absorption spectrum? A graph that shows efficiency of pigments to absorb different wavelengths of light. So definitely pigments are able to absorb light or not. So first one, actually, absorption spectrum. We'll discuss this one again. Absorption spectrum, a graph that shows ability of pigments to absorb different wavelengths of light. That is a absorption spectrum. So this one is a action spectrum. So Engelmann prepared a first action spectrum that is almost similar to absorption spectrum of chlorophyll A. Okay. Next.